Let us now move on to immunosuppression. It is nothing but the suppression of the immunity. They are used mainly to prevent and treat the transplant rejection, idiopathic thrombocytopenic purpura, myasthenia gravis, systemic lupus erythematosus, rheumatoid arthritis, ulcerative colitis, and hemolytic anemia. Immunosuppression happens in two mechanisms. First is cell mediated immune response, second, humoral immune response. Moving on to the cell mediated immune response. This mainly has, suppose, antigen. Now, there is something called as APC, that is antigen presenting cells, which involves macrophages and dendritic cell in case of cell mediated immune response. Now, these macrophages or dendritic cells, they will engulf the antigen, okay, and they will break it down and show to the T cell. Here there is T cell, you can see it is showing to the T cell. Now, there are receptors called as CD4 receptors, and TCR will recognize it, and they will communicate with the chemical called as cytokinins. First is interleukin 1 and interleukin 2. This interleukin 2 will activate the autocrine signaling of T cell or paracrine signaling of NK cells. Not only that, they will also activate interleukin 4, interferon alpha. So, this interferon alpha will act on macrophages and again activate it. This interleukin 1 will convert into helper T cells. Next, interleukin 4 and interleukin 5 it will act on the B cell to mature in the plasma to activate humoral immunity that we will see later. Let us see two major drugs, that is calcineurin inhibitors, immunosuppressants, that is cyclosporin and tacrolimus. They will act on this interleukin 2. That means they will inhibit that interleukin 2, the show mechanisms. Moving on to the humoral immunity, that is nothing but it involves the blood. Humoral is nothing but the blood. Okay. Now suppose there is a bacteria, and this bacteria has something called as antigen determinants, or you can say epitopes. Here the major cell is involved as B cells. Okay, so these epitopes are being recognized by our immune system as a foreign body. So now these B cells in our immune system will recognize them. Particular B cells have particular type of antibody on it. So there is membrane antibody present. So it will be specific, that antibody will only bind to the specific epitopes present on this bacteria. Okay, so when it binds, what happens? The B cells will get activated when it binds here. You can see there are clones of B cells being formed for the future defense purpose. Or what happens, these B cells, when they are getting activated, they form something called as plasma cells or effector cells. They will produce many such kind of millions of antibodies, that, or you can say immunoglobulins, which goes and coats the bacteria. After it coats, what happens? It will be neutralized, and the multiplication of the bacteria will be hampered. And this is how humoral immunity works in our body.